Sorry, it must be my internet. Everything appears to be super slow right now. Okay. Okay, so glycolysis is our first part of cellular respiration, specifically glucose metabolism. So we're talking about breaking down glucose. And our main thing is that we're trying to get energy out of this process of metabolism. So we're trying to make ATP at the end of all of cellular respiration. And to do this, we're also, throughout this process, going to be seeing this molecule NADH, an electron carrier. So it's kind of going to be carrying a little bit of energy for us in a way through the process of metabolism, even though it's not our cellular form of energy out in the world. When we talk about aerobic respiration, we're saying that it uses oxygen. We do also see some cases of anaerobic respiration, specifically a process called fermentation. We're going to see that the cell has ways to make energy, make ATP, even without oxygen present. Okay. So the purpose of this course, we're talking about glucose. Other organic molecules could be involved. Okay, so the part I want to make definitely sure that we all know, right? Our goal is ATP. So hopefully you will know several things about metabolism by the end of this course next week. But if you only know one thing about metabolism by the end of the next week, what I'd like you to know, right, is that ATP has energy, and know where in these various steps we make ATP, because that is the most important thing, the most important part of this process. Okay, so ATP is this molecule, and it essentially holds energy in the bonds between its phosphate groups. So in red here, we have some old sort of tilde signs. These are covalent bonds between our phosphate groups. And when we're saying we're making ATP, what we're basically saying is that we have this molecule, ADP would be this part, and we can tack on an extra phosphate group, which turns it into ATP. This is because this is adenosine triphosphate. Adenosine diphosphate would be a DP because it would be di. Di means two. Okay. So in ATP, we're adding on another phosphate, and we can think of this bond that holds the phosphate group on as storing energy. So through the process of metabolism, we're trying to take energy held in the bonds of glucose and store them in the bonds of ATP, basically. Okay. So the other thing we needed to know about in order to understand all the stuff we're talking about in metabolism is this idea of a redox reaction. So in a redox reaction, what we're saying is that we're taking an electron from one molecule and adding it to another. We call it oxidation if we remove an electron. We call it reduction if we add an electron. So in this case, A is oxidized as we go from this A with an electron to this A without an electron. So this one would be the oxidized form. B gained an electron. So B here is the reduced form. So when we're talking about this in cellular respiration, we're talking about NADH usually. So NADH is going to undergo oxidation reduction reactions, redox reactions in many parts of this process to create sort of energy intermediates, which just means it's basically going to be taking electrons, carrying them around, carrying them to the end of this process, where we're going to see they're going to play a big role in creating a lot of ATP kind of all at once. Okay, so NAD is the reduced form, so we can think of it as having more electrons in it. Okay, and we'll talk a lot more about NADH really on Monday when we talk about the TCA cycle, but we are going to see this molecule in glycolysis today. Okay. So shorthand, I think the video did say it, but when we see an H here, all right, we can think of this as being the reduced form, as kind of the electron being held somewhere in here. That's a biology kind of shorthand. Okay, so the rest of this PowerPoint is just pictures of what's happening during glycolysis, which is the first part of glucose metabolism. So this is the equation for all of glucose metabolism. This is showing you where in the cell we are, because it's going to be really important for our purposes to know where we are relative to the different parts of our mitochondria. Right, so in glycolysis, super important to know you're out in the cytoplasm, because glycolysis can happen even in cells that do not have mitochondria. So this can happen basically in all types of cells, all forms of life. Right? Remember, like our prokaryotes wouldn't have mitochondria, but they'd still be able to do glycolysis. Okay. So this breaks down the three phases, and this is what you're doing in your problem set. So I'm not going to speak about this much because you're going to kind of run through it right now. But basically, we got a part where we put in ATP to kind of prep this stuff. We then break a 6-carbon molecule apart, and then we get some ATP out of it, and we sort of charge up some of these electron carriers that are going to go continue in our process. So cleavage is we're breaking our molecule. Energy liberation is where we're getting the ATP out of these processes. Okay. And here we have our equations. So this first unsimplified equation, when we say unsimplified, I mean that it shows us everything that we put in and everything we got out, but we can simplify it by crossing out anything that appears on the left and on the right, which is why we cross out 2 of the ATP here and are only left with 2 on the right-hand side. Okay, so you're basically just going to be recreating this in your problem set, essentially, with the help of those pictures I've provided to you. Okay, so I will send you to breakout rooms while I try and fix my speakers, and I'll come around. And if they don't get fixed, we'll just use the chat. Actually, you know what I'll do? Hello, hello.